I hope I'm audible. Am I audible? Hello, am I audible?
Yes.
All right, thank you. So in today's class, we will start and as well as finish one small topic. Today we will discuss about stem cells. Okay, what are stem cells? How do you actually know that a cell is called a stem cell? How do you know these stem cells exist? And we will also see some mathematical models that is used to describe their division, their stem cell division. Okay, so let's begin. So stem cells are the source of all mature cells in the body. Mature cells means the differentiated cells. Okay, the differentiated cells, they arise from the stem cells, and there are basically two types of stem cells that are known to us. One is the adult stem cells, another is the embryonic stem cells. We'll describe them in detail. Okay, and some experiments like somatic nuclear transfer. So this is a experiment where a somatic cell means a differentiated cell. So from that cell, people take out this nucleus. Okay, here this red color is the nucleus of the somatic cell, and an egg cell or oocyte, that is the female gamete, they take it and they remove the nucleus of this oocyte. So what they do, they add the nucleus of the somatic cell into the egg cell. So it created a new kind of cell where the content, the cellular cytoplasmic content is from the oocyte, whereas the nucleus is from the somatic cell. So what will happen then? So whatever genetic information that is there in the nucleus of the somatic cell, that will direct the further division of this oocyte, correct? And that is why it like creates clones, means through rapid cell division, clones are created. Now these clones can be implanted into a surrogate mother, or you can do or you can culture these clones using appropriate tissue culture conditions. Okay, in vitro. So this process is called somatic cell nuclear transfer, and in this way we can actually produce a large number of pluripotent cells, means cells that can grow and differentiate along multiple cellular lineages. Okay, and so this is a technology that has developed to create cells with pluripotency. So which is why means the use of the stem cells in the field of tissue engineering is becoming more and more exciting for the people. Okay, now first understand what are stem cells. We have to first so you may be given you may be handling multiple types of cells, and you need to ensure the uh, unique properties of each of this cell line. So you have to know what is a unique property that makes a cell a stem cell. So stem cells are those cells that can self-renew or have the property of clonality, means it can create clones with what are clones, means cells with the same exact features as itself. Like you are familiar with how the amoeba divides, right? It like breaks apart and form two daughter cells which are exactly identical to the parent cell. That is clones are created. So stem cells can self renew, means stem cells can create clones of itself. Then these cells have extensive proliferation capacity, or they have replicative capacity, means they can divide a large number of times to create a large number of cells. And each of these cells, they have the potential to differentiate along one or two lineages. So that this property is called potency. So it can be it can differentiate along one lineage, means it can only differentiate to give say bone cells, or it can differentiate and give multiple types of differentiated cells like muscle cells or cartilage cells, okay. Or it can be totipotent, means it can differentiate along uh, all the lineages, all the known lineages in our body, and create all types of cells in our body, okay. So if I were to explain this in other words, stem cell is a clonal, self-renewing entity that is multipotent, okay, means multiple lineages it can differentiate along, thus have the potential to differentiate along multiple lineages. Now, historically, it has been thought that stem cells can divide without limit, okay, means at least for a lifetime, and produce a large number of cells. So, you will realize how this statement is not exactly wrong, but also not exactly correct. So as more and more research is coming up in this area, we are learning new things about this particular cell type. So as I already told, depending on the potency, means how many lineages the stem cell can differentiate along, you can categorize the stem cell into three types. One is unipotent, it can produce only one cell type. Pluripotent, it can produce many cell types. And totipotent, it can produce all the cell types in our body. Okay. So stem cells are also known to reconstitute a damaged tissue and maintain normal tissue function. So as you already know, the goal of a tissue engineer is to create a tissue engineered medical product that can act as a replacement, or it can act as a therapeutic way to repair the damaged area, or it can improve the tissue function, right? So for these purposes, we sought after making an artificial tissue. So now if we incorporate stem cells in our artificial tissue, it will actually help us to achieve those functions. Now, how do we know the stem cells exist? So what led, or what kind of research in the past have led to believe that such kind of cell population exists in our body? So few example of those research is given in these 3 points. First is people saw that in lethally irradiated mice, means they were exposed to very harmful wavelength of light, say gamma rays, okay. So because of which their bone marrow, or the function of the bone marrow was compromised, and the hematopoietic failure was observed, means no new blood cells was produced. But their hematopoiesis capability was restored when a half dozen of selected stem cells was injected into them. So in this way, you can think that these stem cells are reconstituting, means completely forming new bone marrow in the mice whose bone marrow was destroyed. And because this bone marrow is formed, the new blood cells started to form in this mice. So that actually supports the role of multipotency that the stem cells have, as well as the extensive proliferative capacity and clonality. So these, due to these properties of the stem cells, people have observed this result in this experiment. Another similar experiment is people introduced few genetically marked mesenchymal stem cells. So how can you genetically mark them? You can incorporate a GFP protein, like GFP stands for green fluorescent protein, or you can so if you use a fluorescent protein, you can see under fluorescent microscope where the cells have is present, okay, because this protein gives a green light under when irradiated with a particular wavelength. So you can basically, you have to just insert the gene encoding for the GFP protein, and these cells will be uniquely identified from the rest of the cells. Or other ways of doing can be it will start secreting special kind of a protein, right? Say some antigen or some kind of an enzyme that will be identified through if it is enzyme, then you will identify it through some reactants that gives off sick color, okay? Or if it's an antigen, you will use special antibodies unique to this antigen, and then through antigen antibody reaction you will be able to find those cells. So since it is genetically marked, the gene is present in the nucleus. As the cell is dividing, the daughter cells will also have these genes with them. So because of this logic, people found that uh, the mice which was sublethally irradiated, means the extent of damage that the radiation caused is not that much. So in those mice, they added these genetically marked mesenchymal stem cells, and over a long period of time, when they observed how these cells are, uh, acting in the body, whether or not it is contributing to reconstitution of some organs. So when they monitored it, they saw that the daughter cells of these mesenchymal stem cells can be found because they were genetically marked. So this supports the fact that these stem cells, they can help in reconstitution of damaged tissues and organs. And another experiment, what they did, they like took some mice, all right, and they implanted some diffusion chambers, means there is a membrane, okay, and one side there is high concentration, there is low concentration, so there will be diffusion. So this kind of mini chambers they've made, and they implanted it into the body of a mice, okay. These chambers, they were filled with bone marrow cells. Bone marrow cells, I mean the mesenchymal stem cells and the hematopoietic stem cells. They fill these chambers with these cells, and the pores of the membrane was having a pore size of 1 micron, right? And then they monitored for 3 weeks, okay. So this was implanted into the body of a mice. So what they saw is, after 3 weeks, within these chambers, bone and cartilage started to form, all right. So this statement somehow got reducted, but what I'm trying to say is, the markers, the unique markers of the hematopoietic stem cells and the mesenchymal stem cells, they have started to lose their expression, means their expression declined over 3 weeks, whereas the markers for the cartilage or the bone, okay, so their markers increased. Now, since the membrane was having a pore size of 1 micron, and the size of the cell is around 10 micron, so they knew for sure the bone cell or the cartilage cell could not have entered into these chambers and did this. It is only the bone marrow stem cells that have produced this kind of structures on their own, given that in vivo they are getting the necessary conditions, okay. So the amount of hematopoietic markers in the chamber, they declined, whereas the alkaline phosphatase signal, which is a marker for bone, okay. So bone cells like osteocytes, osteoblasts, they secrete an enzyme called alkaline phosphatase, okay. So that is a unique feature of found only in bone cells. So this enzyme started increasing, secretion increases. Similarly, the cartilage, it also has some specific markers they tracked it. So from this experiment, they concluded that due to the small pore size, the newly formed bone and the cartilage, they had formed only because of the implanted bone marrow stem cells. So in this way, they showed that these cells are having multipotency, means they can differentiate along multiple lineages. Okay, so these are the few experiments that prove the existence of stem cells, or you can say, since people initially found the stem cells to be present in the bone marrow, and you know the bone marrow is the most proliferative organ in the body, and later on people are and now people are still finding new cells, new stem cells in other tissues as well. So whatever early research have happened, they have all happened on the bone marrow cells. All right, so these investigations led to establishing two hypotheses. First is, in the bone marrow there are some hematopoietic stem cells that can differentiate along multiple lineages, and also they persist for a long period of time in vivo. Means these cells, they do not lose their identity very quickly, rather they stay inside the body for a long period of time. So from this explanation to you may be able to understand now what are exactly stem cells. All right, so any queries uh, up till now? No. All right, so how do you know means how do the stem cells look like? Another question. So you're under the microscope, you're observing the cell population, and from morphology only you want to uh, say that this is a stem cell, whereas the other is not, another cell population is not. So we have to know how the stem cells look like when observed under microscope. So when you do antibody antigen interaction kind of studies, where you are going to track some unique cell surface markers, so those experiments, those are very expensive. Okay, and they will not help you for daily monitoring of a cell population. It is the morphology of the cell that tells you uh, at what stage of cell differentiation or division it is. Okay, or whether the cells are healthy or not, whether there is some contamination. So these kind of understanding will make you a better, or make you more skilled in culturing sophisticated primary cells. Okay, so for example, hematopoietic stem cells. So as I told you, most of the research has been done on either hematopoietic stem cells and mesenchymal stem cells, and now only these two cells, these two stem cell types are very much easily available all over India. Okay, not only abroad, all over India, and their cost is also not that much. There are labs in India who are themselves isolating these cells, or you can easily purchase it as at a much lower cost when compared to other kind of stem cells. Because as I told you, a lot of research has been done, and more research is coming up using these cell population. That is why a classic example of how the stem cells look like is coming from the hematopoietic stem cells. Now when people have isolated these stem cells, they identified it using the cell surface markers CD34 plus and CD34 minus. All right, and these stem cells, they are small and spherical looking, and their diameters are also smaller. Okay, around 6 to 8 micrometers, and have no particular morphological features, means you cannot see some kind of pseudopodia etc., though they do have a high nuclear to cytoplasmic ratio. Means the nucleus covers majority of the cell volume than the cytoplasm, and they appear to express receptors for most of the known hematopoietic growth factors, means they can respond to these growth factors easily. They appear to divide rarely and may commit to growth and differentiation in a stochastic manner. So we will see how the cell decides that whether they have to divide or not. So mostly inside our body, the stem cells remain in dormant stage until and unless the tissue is damaged. Okay, so when tissue is damaged, you need a large amount of new cells to come up and fill up the gap, right? So for meeting this requirement, then only the stem cells become activated and start proliferating. So this is an observation that people have reported in their research. Maybe new information will come later on, but that is what I know. So the division is very rare. They hardly divide, and even if the division or differentiation is going to happen, it happens in a stochastic manner. Stochastic means you cannot say for sure what is happening. It is completely luck, by chance. Okay, that is the meaning of stochastic. So one data that is given is one stem cell can divide per 6 weeks. So this number can vary. As I said, it's a stochastic thing. It might be one, it might be two. You cannot predict it very easily. There is no fix. It is not fixed in nature. It is random completely. Okay. So this kind of behavior is shown, or this kind of morphology, like small spherical size, is seen for the hematopoietic stem cells. So other cells like mesenchymal stem cells or neuronal stem cells or embryonic stem cells, their morphological features will vary. Now, which tissues have stem cells? Another very important question actually. So you know already that tissue undergoes uh, for any tissue you can define three dynamic states, right? The homeostasis, tissue repair state, and tissue formation state. And during tissue repair, we have, like, during homeostasis state, we have discussed about tissues like the bone, the skin, the small intestine, where the epithelium layer, like in skin and small intestine, is completely being removed after few weeks or few months, correct? Only to get replaced by a new layer of epithelium. So there is a continuous replacement, a cycling of the where the old cells are removed by the body and the new cells take their place. This is the replacement time, and each tissue have their own replacement time. For skin, it is around 28 days, where the liver, it takes more than a year to become completely a new liver, where the old cells, the old hepatocyte cells, are discarded, and the new cells, they come in and maintain the function of the liver. So for doing this, for from where the new cells are coming, it is coming only because a stem cell system exists in these tissue types. Now, uh, this seems to be true for almost all organs in our body. People proved it for bone marrow, skin, small intestine, okay, muscle. But now the organs which has a very slow turnover time, like the liver, I said, it takes more than a year to for removing the old cells. Means the turnover time, means turnover means the rate at which the old is discarded and the new is formed. That rate is very much slow. So even in those organs, people have found the some very small population of stem cell exists, like in liver, brain, and even the beta islet cells in the pancreas. But the turnover is very small, very small. Maybe one cell, one stem cell will become active and start proliferating in a year, whereas in skin it is say one or two stem cells started proliferating in every 1 month. So this kind of rate varies. But yes, stem cell populations is being discovered in almost all body type, all organ and tissue types in our body. Now, what fraction of adult tissues are stem cells? Means out of, say, in our organ, around 10 is to per 11 cells exist, if you remember the numbers we discussed in the previous classes. So in those 10 is to the power 11 cells, how many are those are the stem cells? That is the question basically. So to know that, we have to understand what is written in this few points. It is important to know that the cells that is present in the adult tissues, means the tissues of our body currently, they all have a finite lifespan. means they will die soon. Whereas the stem cell population, they remain dormant. They don't work until and unless it is required. So our organ cannot give them a very large space for this dormant cell population. Otherwise, the organ function will stop, right? So that is why each organ has a very small population of stem cell. Like for example, in bone marrow, 1 in every 1 lakh of cells is known to have a stem cell characteristic. So that is why it is very difficult to correctly isolate them and then characterize these cells. Because you can understand, in 1 lakh cells you have to find one cell, and only that cell will have the stem cell like characteristics. So it's a very difficult job to do it, and then further work with it in vitro. So yes, that is exactly what I said, that this led to the attempt to isolate the stem cells for tissue engineering, and since they are the only cells that contribute to long-term tissue maintenance. Okay, so now I told you initially of the initial point of this lecture that there are two types of stem cells. One is the adult stem cell, another is the embryonic stem cell. So what are these? Which stem cell is called adult stem cell and which is called an embryonic stem cell? So if in between any one of you has any query, you guys can like freely speak up and let me know. All right. So the adult stem cells, they are found in the adult body, okay, and they are thought to be tissue specific. Adult means the baby is born, right? It is outside in the world, and the cells that are there is fixed, right? And inside the tissues, there is a small population of cells. Now these cells will be now called the adult stem cells. Like in bone marrow, the hematopoietic stem cells, the mesenchymal stem cells, these are all adult stem cells. Okay. Whereas the embryonic stem cells refer to the cells that is derived from the early embryo. So you remember the structure of an embryo, right? Means the steps in the embryogenesis that we discussed previously. So, and you remember that like the cells, the fertilized egg or the embryo, it divides, okay, divides rapidly into 2 cells, then 8 cells, then 16 cells, right? So 16 cell stage is called a blastocyst, okay, and the blastocyst consists of two components or two type of cell population. One is called the inner mass cell and another is the trophoblast. So these things I hope you remember because we have already discussed. So people have found in a few of the inner mass cells, okay, they are the embryonic stem cells, okay, because these embryonic stem cells, they can basically form tissue from any of the germ layers like the endoderm, ectoderm or mesoderm. And these cells, they are thought to be pluripotent, you can say almost totipotent, because they can give rise to the cell population from all the 3 germ layer types. So if I were to ask from where the embryonic stem cells come, so you should be able to immediately answer that they are present in the inner mass of the blastocyst structure of the embryo, okay. Now again, we have also discussed the term plasticity. Now this term again is coming to us, okay. So let's understand what is now stem cell plasticity. For many years it was believed that once a stem cell is committed to a certain tissue type, it becomes restricted to producing progeny of that same lineage only. Means you can't expect a stem cell found in skin, say, will produce bone cells, okay. This was the previous previous belief. But what is plasticity? Plasticity means it can give rise to any lineage. Okay, in this context, here the plasticity means this. So it is thought that only the embryonic stem cells could differentiate into the cell types of the different germ layer. Means complete plasticity is shown by only the embryonic stem cells. But now the recent research that, as I told you, it's a very new field. More and more labs all over the world are studying these type of cells. So they are now indicating that the adult stem cells, which are found in a specific type of tissue, they do retain, or they do have the capacity, or they do still have the capacity to give rise to cells of different lineage when it is placed in an appropriate microenvironment. Means you have to give the proper conditions for this plasticity behavior to happen. So for example, people have shown that the hematopoietic stem cell, which is an adult stem cell, it can actually in fact give rise to hepatocytes, myocytes, or neurons. Okay, but in fact, what happens inside our body? These stem cells are responsible for forming the blood cells, but in vitro, when appropriate condition was provided, they could form the liver cells, muscle cells, as well as the neuron. So this is called plasticity or stem cell plasticity. Okay. Now, so far, is there any query related to anything that I have discussed? Because now we will move on to a slightly different topic. All right. So there is no queries. So am I audible clearly to you?
Yes, ma'am.
All right. So far we basically had an introduction on what is stem cell, which tissues have stem cells. Now let me describe what happens at the molecular level of these type of cells, which basically underlies very big question like why we undergo aging, why do we age, and why do we have a limited lifespan, or cellular level, why the cells have a limited lifespan. So in the discussion from now onwards will help you understand those, or help you understand the answer to those questions. So do stem cells really self-renew? So if you remember, self-renew means clonality, clones. Two things are clones if they're exactly identical. So do they really, when the stem cells divide and form new stem cells, are they exactly similar to each other, or does something happen, or does something changes at the gene level or at the chromosomal level? So we will understand that now. So I hope you all are familiar with the process of DNA replication, correct? Yes or no? Everyone is aware of how the DNA replicates. I'm not going to ask, just a simple yes and no would work, so that I would know that whether or not you'll be able to understand. Yes, Vaishnavi, January, you guys unmuted and then again muted yourself. So DNA replication you guys know, right?
Yes, ma'am.
Yes.
Okay, correct. So what happens with DNA replication? when the DNA polymerase acts on the template DNA, it creates two new strands. One strand is formed continuously, it is called the leading strand, and another strand is called the lagging strand. So the in the lagging strand, you know, Okazaki fragments are formed, which are later on ligated by the DNA ligase. So now, due to this formation, or the way the lagging strand forms, what happens is the synthesis of the last Okazaki fragment, it cannot be initiated with DNA polymerase. Means when the Okazaki fragment is formed from the 5' to 3' end, and the complete template is being read in that in the opposite direction, the last few base pairs of the template, they are not exactly copied to the lagging strand because the DNA polymerase cannot act there. Okay, it is maybe because suitable RNA primer cannot be formed, so which is why the DNA polymerase cannot act and cannot form the last Okazaki fragment. So because of which the lagging strand will be always shorter than the leading strand if you compare the number of base pairs in the lagging strand and leading strand. So what happens in that case? The DNA, the newly synthesized DNA, will be slightly shorter than the, or few base pairs shorter than the parent DNA or the from the template DNA. Correct? So you also must be familiar with the structure of a chromosome. And if you recall, this chromosome, when it is in uh, homozygous chromosomes, when they are formed, so their center is the centromere that connects the sister chromatids, and the ends of the sister chromatids are called telomeres. Correct? And the telomere, it contains majority of the non-coding repeating sequences. Means the sequences don't code for anything, they are just repeats, like ATGC, like repeating 100 times, 50 times. Okay, so these ends, the telomeres, it does not contribute to any genetic information, they are just non-coding repeating sequences. Now, due to how the DNA is synthesized, where the lagging strand is becoming shorter. Okay, now if this thing is going to happen throughout the chromosome, so what will it will contribute? It will contribute to shortening of the telomeres, length of the telomeres. Okay, and as it is shortened, okay, the, you can say the cell is aging. The shorter the telomere, the closer it is to senescence or the old age. After senescence, the cell division will stop, and it is also the time when our body basically goes towards decaying, dying, right, in old people. So at the cellular level, this shortening is called the mitotic clock. So normally, this kind of behavior is seen in the somatic cells, means the differentiated cells. It is not seen in the gametes, the female and the male gametes, the sperm and the egg. So they don't follow this mitotic clock behavior. So, and people have observed that per replication, means per doubling, there is around 50 to 200 base pair loss in the telomere. Okay, so in every replication, the telomere length is getting shorter by 50 to 200 base pairs. So at the time of birth, the length of the telomere is around 9 to 11 kilo base pairs, and till we reach around old age, the length of the telomere reaches to 5 to 7 kilo base pair. And at this level, when the telomere is at this length, the replication basically, cell replication basically stops. Okay, the structure of the chromosome, it becomes unstable, and the cell starts to die, as it is not going to receive the necessary survival genes, or the expression of these genes won't happen which is going to dictate the survival of the cells, and basically cell will start dying. Okay, so you now have the questions of how the cells age, or rather how do we age, or what happens at the molecular level that leads to aging of a cell. Is because of the telomere length, how it is decreasing with every replication. All right, and how telomere length is decreasing? It is because during DNA synthesis, the lagging strand is shorter than the leading strand. All right, I hope you have understood this part. All right, is it yes or no? Any query from this part? Any
No, ma'am.
all right, great. So this mechanism, the previous mechanism, the mechanism that I discussed in the previous slide, I'm talking about that mechanism. It basically is the underlying reason of why the so-called Hayflick limit exists. If you remember that cell that is isolated, say from the fetus or from a newborn baby, it can go at least 50 to 30 doublings, right, before it reaches cell senescence. So this kind of Hayflick limit is coming into place because of the molecular event that I have discussed so far. So cell telomere shortening is not the, of course, means telomere shortening is just one of the events that contributes to complete cell cycle arrest, but it is one of the main events that ultimately starts a domino effect. एक शुरू हुआ, then arrek means it just start of like inevitable cell death. Okay, so culture conditions means here I'm talking about in vitro, not in vivo. So in vitro, if we give correct culture conditions, then we can actually either have a positive effect or a negative effect. Means that is how people have established continuous cell lines, right? We discussed when we are talking about tissue culture that there are continuous cell lines that continuously grow for more than 50 to 50 doublings, because the culture conditions they were grown, it actually helped them to surpass this limitation. Whereas if the culture condition is not appropriate, then the cell will start dying even if 50 doublings has not reached. Now the telomeres, they can be rebuilt, means you can replenish the lost length in the telomeres by using an enzyme called telomerase. So the activity of this telomerase is seen in microorganisms like the yeast. It is also active in the germ cells like the egg cell and the sperm, and it is also active in a majority of human cancer cells. So now you can understand why these type of cells are able to basically maintain their population for years, because this telomerase enzyme is acting, that is constantly replenishing the lost telomere length. The gene for these enzymes, it can be inserted into cells to increase their lifespan. So you can do it using a plasmid vector or a virus vector or BAC, etc. You can do it. The telomerase is a basically ribonucleoprotein DNA polymerase that elongates telomeres in eukaryotes. So it's a ribonucleo DNA polymerase protein. Now, do stem cells really self-renew? We are continuing with that. So when telomerase is expressed, it is going to maintain the length of the telomere. Telomere hypothesis implicates short telomere length and telomerase activation are critical players in cellular immortalization and cancer. So if you remember, immortalized cell lines are the continuous cell lines. There the telomerase is active, so because of which the telomere length is not lost, rather it is continuously being rebuilt. All right, and also in cancer cells, they can proliferate continuously for n number of doublings because this telomerase gene is acting there. Now, strictly speaking, stem cells cannot self-renew without limit. So now you can understand that the stem cell clones that are going to be formed inside our body, or say in vitro, they are not exactly clones, because the telomere length is going to differ, say by 50 base pairs from their parent cell to the daughter cell. Okay, and also it poses a limit up to how many times it can divide and create new stem cell daughter cells. Okay, so telomerase activity, it has been found in hematopoietic stem cells, but the activity is very low. So if the high activity is there, then you means the stem cell population is will be like infinite, right? There will be no shortage, there will be no death. So if this kind of basically research paves the way for the research like which prolongates human life, okay, eradicates diseases, but you cannot do it unlimited. As you know, cancer will start developing, because in cancer telomerase activity is the highest. Okay, thus although there is evidence that telomeres in immature hematopoietic stem cells do shorten with donor age and with increased cell doublings in vitro, the rate of telomere shortening in stem cells is finite and may be slower than in somatic cells. All right, so telomerase is there, or activity of telomerase is seen in stem cells, uh, and but the activity is less in comparison to those found in cancer cells, or as well as in normal cells, because of which uh, the length of the telomere is not that much reducing as it is seen in somatic cells. Okay, now we will discuss how the stem cells are being used or for tissue engineering purposes, or you can say why should we consider using stem cells for tissue engineering. Now we will discuss that aspect. Stem cells in our body, they contribute to homeostasis, right? They contribute to repair, they contribute to organ function. So similar kind of, if you want to mimic similar kind of functions, we can use stem cells, correct? In artificial tissues, cellular fate processes of cell proliferation, cell death, cell motion, cell differentiation, they are all the cellular fate processes that determine the fate of a differentiated cell, ultimately the fate of a tissue. So if the stem cell population in our organ or our tissue is lost somehow, then the organ function is partially or completely lost. Say if in bone marrow, you have seen in the mice model, the bone marrow was irradiated, because of which the hematopoiesis stopped, right? No new blood vessels started forming. So in that is what is understood from this statement. So the stem cells, they can be thought as a starting material for making new tissues, all right, and they can be also thought of a source through which we can regenerate the damaged tissue in vivo as well as in vitro conditions. So that is a reason behind why stem cells is being considered for tissue engineering applications. Stem cells for the basic tissue types. Now, you know that say human hepatocytes or cardiomyocytes, these cells, they do not grow well in culture, and for making artificial tissues you need to culture them so that they grow to a relevantly large number of cells. But suppose if you were able to signal a stem cell appropriately in vitro and uh, move its differentiation along the lineage that gives rise to hepatocytes or give rise to cardiomyocytes, then your job will become much more easy, right? Because stem cells will first proliferate a lot in number, all right, and then the proliferated cells will give the differentiated mature cells. So similar thing people usually do. Uh, Whatever tissue that people have to repair, you can categorize them or into either connective tissue type or an epithelial tissue type, which you already know that we have discussed during the previous classes. The epithelial tissue, how it differs from the connective tissue. So in connective tissue, what are the stem cells that is available for the connective tissue? One is the hepatopoietic stem cells, another is the mesenchymal stem cells. Now both of these stem cells, they will differentiate along the lineages that give rise to connective tissue cell types like the blood cells, bone, cartilage, etc. And these cells reside in the bone marrow. So you take out these cells simply, give proper conditions and make bone cells or muscle cells, etc. Okay? In vitro or directly into your artificial tissue construct, if you can do it, then that will be much more beneficial. And for epithelial tissue types, people are starting to discover the stem cell population that is present in those epithelial tissues. Okay, so first we have to isolate those epithelial tissue stem cells. Okay, and then do the same process. Whereas for organ like liver, as I told you, liver has a very slow turnover rate. So for liver, how will you use the stem cell? So people have observed that you can actually induce the plasticity, as I just told, stem cell plasticity in hematopoietic stem cells to make liver cells like the hepatocyte cells. Okay, so for as a starting material, or depending, since we know a lot about the hematopoietic stem cells and the mesenchymal stem cells and their potential to form different cell types, that is, and also undergo plasticity, that is why it is being written that the bone marrow can be thought as an attractive source or the stem cells through which you can create new tissue types. That is why more and more research is being done in these two stem cell types, and for different organs. Okay, for different organs like the brain or the nervous tissue or the eye corneal tissue. Okay, so for all these type of tissue types, the skin, so the mesenchymal stem cells or hematopoietic stem cells, their application is being shown. Okay, now another challenge that comes is isolating these cells. All right, that is a very challenging thing to do, because like I said, in bone marrow, once is out of 1 lakh, that is the fraction of the stem cell that is present in bone marrow. There is one stem cell in 1 lakh cells, so you have to uniquely identify that one cell. So in order to do that, people have used affinity columns or magnetic columns or counterflow electration. So there, what they do, they either use antigen antibody interaction, where antibodies are present in the column. Okay, and these antibodies are designed to uniquely bind to specific stem cell antigen. Okay, and through that, when they pass the cell suspension through this column, only the stem cell having that unique antigen will bind, rest will flow out. So this kind of logic is being used to uniquely identify the stem cell population. Okay, so difficulty in positively identifying the presence of stem cell is basically the first thing to do. Okay, then only you can work towards constituting artificial tissue that will go inside your body and help in reconstitution of organ and tissue, that is repairing the damaged tissue. So that's the first thing that any tissue engineer has to think about. Now, ex vivo growth and manipulation of stem cells. Ex vivo means you have taken the cells out of the body and growing it. Okay, that is ex vivo. So you can grow these cells ex vivo, but it is a bit difficult. All right, because what I have observed in our lab is that these stem cells, after few passages, after few number of times of passages, they start differentiating on their own, even if we didn't supply anything different to the culture media. Okay, so it is difficult to grow them without differentiation, because they have a tendency to undergo differentiation quickly in vitro, not in vivo, in vitro. Okay, and also the adult stem cells, they age, means the activity of the telomerase enzyme, it is not that much, because of which the length of the telomere is reducing. So extensive proliferation cannot happen. Okay, it has a limitation that bounds this limit, bounds the number of doublings the stem cell can undergo. And if we talk about the embryonic stem cells that is taken out from the blastocyst, they can also be cultured in vitro, okay, like in tissue culture conditions. However, we are not completely aware of what differentiation cues are needed, or what kind of growth factor signaling, etc., is needed to signal these embryonic stem cells, or to control the differentiation of these embryonic stem cells into a desired type of lineage. It can happen randomly. Differentiation can happen randomly into a particular type of cell, uh, and we may not able to understand how it happened. Okay, so a lot of understanding is required when working with embryonic stem cells. Okay, because these stem cells can differentiate along any of the three germ cell lineages. Okay, it can form a cell from the ectoderm or endoderm or mesoderm, but you need to know how to control it. Then only it will be beneficial for your application. All right. Now, procedure of isolating stem cells and culturing them for transplantation purposes, it has a number of unique advantages to it, but it is quite difficult. Okay, so once stem cells they differentiate, they will start losing their potential to engraft in culture. Means it starts becoming what do you say, towards senescent stage. It starts going towards senescent stage, and at that stage it may not be very much useful for your purpose of making artificial tissue that you are going to later on engraft on a mice model, a rabbit model, etc. So generally it is what people do is they culture stem cells under conditions so that they do not differentiate, and these naive immature cells, they are used, okay, rather than the differentiated one. And inside the tissue construct, these stem cells, when implanted inside the body, now they can undergo differentiation. But when adding inside the tissue construct, they are added in their immature form, not in differentiated form. Okay, because it is a 3D construct, and there some kind of in vivo microenvironment you will be able to create, and then you will be able to prove that you have successfully mimicked that in vivo microenvironment that have led to the differentiation of the immature stem cell. So this kind of studies or experiments is very much available in the literature today. Okay, so you have to first know what kind of growth factors, nutrients, etc. are needed so that you can culture these cells in vitro for a long period of time without them undergoing differentiation and loss of pluripotency. So that in immature state, you will be able to use them for the tissue engineering applications. So any queries so far related to the topic that we discussed just now, how stem cells are used for tissue engineering, why they are used? Okay, is there any query?
No, ma'am.
Okay, so now we will discuss about few examples of stem cells that are known to us very nicely. Means we'll see them in literature, people are using it extensively. It is easily available, like they are not that much expensive, and a large number of protocols, etc., is available on how to handle these cells. The first cell type is the mesenchymal stem cells. So they reside in the bone marrow. Okay, and these are the cells which can differentiate along a number of connective tissue cell type lineage. So in this example, so in this figure actually, it is shown how a mesenchymal stem cell first proliferate, means a large number of mesenchymal clones are there, and then each of these clones can commit to either become a bone cell osteocyte or a chondrocyte or a myotube or stroma cells, tendon or ligament, fibroblast or adipocytes or dermis, etc. So these kind of lineages or formation of the cells from these lineages is possible from mesenchymal stem cells, given you are giving particular type of conditions that led to this commitment. Okay, so this is the this commitment stage is the important stage. That is the stage where you control or you exert some control over what kind of final cell product you will get from a stem cell. So these cells are found in the bone marrow. They can differentiate to give osteocytes, means bone cells, chondrocytes, myoblast, fibroblast, adipocytes, etc. There was a one recent study that conclusively gave evidence for the existence of these mesenchymal stem cells. So what they did, they isolated these mesenchymal stem cells, okay, and allowed and gave suitable growth conditions, okay, special growth conditions, and saw that these cells have formed cell types like adipocytes, the fat cells, bone cells, chondrocytes. So this conclusively proved, so these connective tissues can only be formed from a stem cell population, which they termed as mesenchymal stem cells. So this is a proofing experiment of how, or proving the existence of mesenchymal stem cell experiment. Okay. Now in the liver also, people have found stem cell population that is called the liver stem cell. Now the existence of this stem cell is controversial. Okay, so many researchers do not believe they exist, whereas some believe they exist. Okay, and how they function, there is also a conflict of opinion. So some people, some investigators believe that hepatocytes, their location is at the sinusoid. Okay, and once they're triggered, they proliferate and differentiate, and they uh, roll down along the sinusoid and they ultimately die. Okay, this is one hypothesis of, or one assumption of what is the location of these liver stem cells and how do they function. So what is a sinusoid actually? Okay, so for understanding what is a sinusoid, you have to first briefly understand the structure of a liver. So in this diagram, you will see this is a liver, okay, and the liver is composed of liver lobules. These are hexagonal lobules, 3D hexagonal structures, okay, and if you closely look the structure of this lobule, it looks something like this, where these uh, pink colored structures with around almost purple color centers, these are the hepatocytes, okay, and these blue colored uh, tubes, they are some veins, okay, there are some passages. The center is the central portal vein, where these veins are coming, basically coming in all 6 directions and joining. Similarly, there are veins, are the arteries that uh, is responsible for circulation of the blood in the opposite direction, and in between these hepatocytes, there are tiny gaps which are called sinusoid. So this is much a closer view actually. And the green color veins, green color tubes, these are the bile ducts. So this is the green colored bile duct. This is the portal vein. It is responsible for delivering the deoxygenated blood. Whereas from the hepatic artery, the red colored tubes where the oxygenated blood is coming. So these are the hepatocytes. And in between the hepatocytes, there is small gaps, small junctions, which is called a sinusoid. This is how the you can see the functional unit. Liver lobule is a functional unit, like nephron is a functional unit of the kidney. Similarly, liver lobule is the functional unit of the liver, and this is how the lobule looks like. So some group of researchers believe that the stem cells, they exist at around beginning of this sinusoid. Okay, like here where I'm putting some dots, and in this beginning spot, these stem cells, they once activated, they differentiate and form hepatocytes. So basically they join the hepatocyte structure, and after few number of divisions, they ultimately die, and it is removed from the body via the veins. Okay, and this is, they believe that it is going on continuously inside the liver. Whereas another group believes that liver function of the liver, or the cell population of the liver, is completely sustained by the division of the mature hepatocytes. Means hepatocytes, mature hepatocytes, they are only dividing and maintaining the cell population. Whereas the earlier proposal was that it is the stem cells which are continuously dividing and maintaining the cell population. But in some case, if the division of these hepatocytes is restrained, maybe due to some tissue or organ damage, then it is the liver stem cells that becomes activated and start cell division and replenish the lost cell number. This is another school of thought that is presented by the people on how the liver stem cells function. So there is no clear consensus like what is correct so far, because a lot of new research needs to be done on this liver stem cell population. Because if you are able to understand how these stem cells function, then you can make liver models, or you can make artificial liver tissues, and that will be highly beneficial for the patients who undergo diseases like cirrhosis, yeah, fatty liver, or complete liver failure. Okay, and need liver transplantation, and liver is a very difficult organ to find. Okay, because of which billions of people are dying. So a lot of research in this area needs to be done. Now, neuronal stem cells. So it is another stem cell population that people have discovered that contributes to the cell types of the nervous tissue or neuronal tissue. So people have isolated this cell from the ventricular zone of the human fetal brain. From the fetus, the brain of a fetus, these cells were first isolated and they were cultured and shown in vitro that they can differentiate into major neuronal cell types like the glioblast, oligodendrocytes, astrocyte, neurons, etc. So they can form basically all these types of cells in vitro. Okay, and when they transplanted these cells back into the brain of a newborn mice, this led to integration. Means these human neuronal stem cells, they successfully integrated inside the brain of the newborn mice and contributed to the overall tissue growth. Because you can definitely identify that by seeing the level of gene expression, because you know some genes will be unique to the humans and some genes will be unique to the mice. You just have to track the expression of the gene that is unique to the human, and then only you will be able to conclude how the integration happened. Okay, so where is ventricular zone in our brain? So again, very simple overview of the anatomy of our brain. So this is the skull, all right. Inside our skull, our brain is there, and inside this pink color tissue which we often think, okay, that is the brain part, very soft tissue, and inside there are ventricles, like these are the ventricles. Okay, and inside these ventricles the neuronal stem cell population was found, and directly in touch with the ventricle is the spinal cord. So there is meninges. Basically this is the tissue that is present in between the skull and the brain. Okay, and there is fluid between the meninges that protects our brain from any shock. Okay, so this kind of structure is there, and inside the ventricles people have found the neuronal stem cells. That is the important thing to remember, the location of the stem cells. So um, so these are the stem cells that are present in the adult body. You can say the mesenchymal stem cells, the hematopoietic stem cells, liver stem cells, neuronal stem cells. These are adult stem cells. Okay, and it is also, you must be also observing that these stem cells are found uniquely at a certain position inside an organ. Okay, it is because body or the tissue wants to, or the organ wants to protect these cells, okay, from external damages, because they are the most precious cell types, and that is why they are present in very small number but occupying a very safe space inside the organ. That is why it is so while isolating the stem cells, we have to first identify where in the organ you can find the stem cells, and it is the first thing to do. If you are able to do it, then isolation of the stem cells become easier. Okay, but you just keep in mind that they are present in a very safe space inside the organ, a very unique space. And in most studies, if any paper you take up where they are discovering some stem cell, first thing they do is report the location of that, potential location of that stem cell population inside the organ. Okay, now we will describe the mother of all stem cells, or mother of all cells from which all the cells in our body have derived. It is the embryonic stem cells. As I've already mentioned, these embryonic stem cells are present in the earlier stage of embryo development, right? And they have the capacity of generating all the cell types in the fetus, and subsequently in the adult. Means the fetus will subsequently develop into an adult. So this is the mother of all cells. That is why it is the mother of all cells. But please note that embryonic stem cells are not totipotent. They are not totipotent. Totipotent means differentiate to produce all cell types. So the totipotency is not shown by these embryonic stem cells. Why? Because these stem cells which are present in the inner mast cell, they cannot function without the role of the supporting cells, means the trophoblast, cells present in the trophoblast, because it is the trophoblast that produce the placenta and the supportive structures. So whatever signaling factors that is produced by this trophoblast, it is necessary for the action of the embryonic stem cells and ultimately create cells of all tissue types. Okay, that is why there is no stem cell that is completely totipotent. Even the embryonic stem cell is not totipotent. The first time in the early 1980s, it was the first time they isolated the embryonic stem cells, and from where they did it? From the inner mast cell of murine blastocyst. So in the mice model, people usually do. So in the mice model, from the fertilized embryo, it was allowed to divide into certain cell number to become blastocyst, and from that blastocyst in early 1980s they derived the embryonic stem cells. And it's till like from then on, people have used these embryonic stem cells for studying embryogenesis, that is the process of development of the embryo, then gene function, and the developmental processes basically. And people have also shown that these embryonic stem cells, they can be differentiated into different multiple lineages, like the neural cells, hematopoietic cells, cardiomyocytes, etc. And they have shown it in vitro as well. And after doing all this, like after proving the concept that it is possible in mice model, people have also now isolated embryonic stem cells from primates and humans. So if the thing is, if we are able to find a source, a continuous source of embryonic stem cells, then it is possible for the tissue engineers to produce or to generate any kind of tissue. Okay, we can basically create any kind of tissue if we can find a continuous source of embryonic stem cells. And also we have to understand how to control the differentiation of these embryonic stem cells into a particular lineage. So these two things we need in order to use or primarily use this stem cell system. Okay. So here in this slide, what it is said that we can use embryonic stem cell derived products, the differentiated cell products, for various clinical applications, like to treat neurodegenerative diseases, diabetes, spinal cord injuries, hematopoietic repopulation, etc. So for these kind of clinical application, we can use these embryonic stem cells. And the use of these embryonic stem cells and their progeny cells, it has opened up a new cell source, right? It has opened up a new cell source, and which is very, very good for making new artificial tissues. Okay. So what people have like started thinking about is creating a bank of embryonic stem cells, all right, and match the MHC alleles on these type of embryonic stem cells to the recipient, so that we can directly transplant these stem cells inside the recipient. Just like we do blood group matching, similarly for tissues as well, people do MHC matching. So for cell also we can do the same, and we can modify these embryonic stem cells or modify the MHC group of these embryonic stem cells to create donor cells, universal donor cells, cells that won't be rejected by anyone. The body won't be able to identify as foreign. Okay. The keys to the development of such novel cellular therapies are like design of culture conditions, procedures that allow for control of cellular fate processes that ES undergoes, and control of cellular organization into tissues. So as I already told you, first we need, in order to successfully or exploitatively use these embryonic stem cells, we need a continuous source of embryonic stem cells, and next we need to understand how to control the differentiation of these stem cells into a particular type of lineage. Okay, and once we have done it, then we'll be able to develop special culture conditions, right, and as well as control cellular organization into a particular tissue type. But as you might have already guessed, that there is a lot of ethical concerns are present inside the research community and as well as the regulators, who because of which we have not completely yet started using these embryonic stem cells for our tissue engineering, for different tissue engineering applications. Very few studies are coming up, no doubt, but most of the studies are from the mouse or for the mouse model, where people are trying to prove uh, some concepts based on embryonic stem cells. Using human embryonic stem cells is a very, very, very big ethical question mark. Okay. So any queries so far regarding the different stem cell types that we discussed? I hope you find this topic interesting, because I recall that before, when I said that we will be discussing stem cells, few of you took interest. So now maybe that interest is being fulfilled. Are you finding this topic interesting, or is just boring? So today, maybe due to the class timing, the people who generally talk are not present. no worries. So after
The telomerase part, it is very like discussion wala part, very controversial there with the telomerase shedding and the aging concept. So
it is interesting, right? I actually couldn't understand the last thing that you said. All right. So I think that was positive feedback. What I could understand from your comment. So I hope others also feel the same about this topic. And after I have shared this lecture content with you all, take your time to go through it, because this week there won't be quiz. Next week there will be. So before that, before the quiz, you guys have ample time to like go through these slides and understand the topic. And if you have any queries, you just let me know, drop a message or send an email, or maybe we can discuss over a call if you want. So these things can be arranged. Now the very interesting topic. Okay, and it is like the most interesting topic to me so far in this chapter. I'm not very much interested in learning the theory. I'm like a person who likes to apply stuff. Okay, that is why I cannot mug. I need to understand. So these few slides, these at least 5-6 slides. After 5-6 slides will be done. These 5-6 slides I find it very interesting. Okay, and there is a example as well, math problem, that I'll do it in front of you guys, but I would also request you to try it on your own if you are able to understand the question. Okay, so how do stem cells divide and what happens when they divide? This is the question that we're going to address in this few 3-4 slides. So people have described the models, few models that actually explains how the cell division, stem cell division takes place. 3 models describing the dynamic behavior of the stem cell population. Okay, so first there are 3 models. First is the clonal succession model, then other is the deterministic model, and third is the stochastic model. Okay, now the clonal succession model, it relies on the fact that, say, one stem cell is activated among the reservoir. So there is a reservoir of stem cell which is generally dormant in a tissue, and one of the stem cell got activated. Then what happens? Then how many new clones, stem cell clone will be formed, or how many differentiated cells will be formed? So that is being addressed in all the 3 models. So for the first model, clonal succession concept, here what happens? The daughter stem cells, or the daughter cells that is formed as a result of cell division, all of them will give rise to mature cells. Means all of them will get mature. All of them will be differentiated to create a differentiated cell. None of these daughter cells will go back and contribute to the reservoir of stem cells. All right. So if this kind of division goes on for a long period of time, then what will happen? The entire reservoir of stem cell will finish, right? And it will be up to the differentiated cells to proliferate and maintain the cell number of the organ. Okay, but also that has a limitation up to how much it can go on. So this is one model of describing how the stem cell division is taking place. Now another model, that is deterministic model, where what happens, this model is based on the assumption that the stem cells can truly self-renew. They can truly self-renew. How? So say one of the stem cells in the reservoir, it got activated and starts dividing and forms 2 cells. So there is a 50% probability that one of the daughter cell will maintain its stem cell characteristics and contribute to replenishing the stem cell reservoir, while the other daughter cell will undergo differentiation along a particular lineage. So there is 50-50 chance of happening a self-renewal, and another 50% chance that the daughter cell will undergo differentiation. So in this way, for every cell division, one stem cell clone is created that is maintaining the reservoir. So if there are say 100 cells, the 100 cell number will be maintained after every division, correct? Because in every division one cell, there's 99 cells will be there in the reservoir and one cell will be lost. But due to deterministic nature of the cell division, the lost cell, that is the one number of cell, will come back. So 100 cells will be always maintained in this way. Now in the third model is called the stochastic model. Stochastic means, as I said, by chance, anything can happen, it is random in nature. So upon cell division of the stem cell, what can happen? Either of the daughter cells means either both of the daughter cells will undergo differentiation, okay, means none of the daughter cell is contributing to the stem cell reservoir, or one daughter cell remains in the reservoir, another daughter cell undergoes differentiation, okay, or both of the daughter cells will maintain its stem cell characteristic and replenish the reservoir, okay. So either you can get 0, 1, 2 stem cells or daughter cells. That is what the stochastic model says. So this figure here you will understand in a summary manner what I have discussed so far. I have discussed about 3 models that has been proposed for how the stem cell division takes place. The first model is the clonal succession model, where upon the division of the stem cell, all of these cells are differentiating. There are 0 stem cell daughters. So the reservoir is completely depleting after every division, correct? And there is deterministic model. Here what is happening? There's 50% chance that one cell will remain in the reservoir and another cell will differentiate. So the stem cell reservoir is being maintained. Total number of stem cell reservoir, it is maintained. But in the stochastic model, means it is happening completely random, okay. There is no like fixed pattern of how the cell division will take place. So in say one cell division, all the daughter cells is going on along the differentiation pathways and differentiating. In another cell division, one daughter cell is staying back, another daughter cell is dividing and differentiating, or another cell division like number in another doubling, what is happening, both of the daughter cells is staying back and maintaining the reservoir. So it is completely random, okay. So you can get 1, 0, 2 daughter cells, stem cell daughter cells, okay. These are the 3 models. Now this is the problem part where I was talking about that I will be doing one small example based on how the stem cells are dividing. So the concepts that we studied so far, the 3 models. So based on that, we can formulate mathematical models to describe the stem cell division or the rate of stem cell division, or what is the probability that one stem cell will remain and replenish the reservoir, means renewal, what is the probability of renewal, and probability of aging, means the stem cell is also aging, right, because telomere length is declining. So based on those concepts, we usually formulate mathematical models to answer or to describe these facts, these numbers. This is the example that is given to you so, in the figure format, okay. So try to understand what is given in the figure, then only I'll come to the questions part. Wait a second, let me drink some water. So if you are having a pen and paper with you, you can maybe take it in your hand right now, so that you can do the problem along with me. Okay, so it's this year column, it's showing the age of a person. Now you can say it is completely proportional to the age of the stem cell. All right, but the telomere length is showing what is the length of the telomere in the stem cell before it is committing to differentiation process. Right, so this is the stem cell that is present inside a newborn baby, that is year 0, and say and once it is born, say due to some event, this stem cell has started to has become active, because of which it forms a progenitor cell. This is the progenitor cell, and this progenitor cell starts dividing multiple times. Here 30 doublings is taking place and giving rise to 10 to the power 9 differentiated cells. Now the telomere length of this differentiated cell is 9.5. So similarly, data is given for different number of years. The telomere length of this stem cell before division is given, and the telomere length of the differentiated cells is given, and as well as how many cells are formed, number of cells is also given. Now note that when this first cell, this one at year 0, when it is undergoing when it is undergoing division, cell division, then one daughter cell is becoming a progenitor cell, whereas another daughter cell is staying back and like maintaining the reservoir of the stem cell. Okay, so there's a deterministic manner the stem cell reservoir is being maintained. All right, and also the rate at which the loss of telomere length is happening in the stem cell and in the differentiated cell, that rate is exactly same. That model also assumes that. Now let's try to answer few of the questions. So I've opened a notepad here. Okay. First question is, after 30 doublings, how much telomere length is lost in the differentiated cells? Okay. So how would you do this question, or how would you understand how to begin, or how will you begin to solve this question? So number of doublings, say n is equals to 30, right? And you have to first look into the initial length of the telomere, means at year 0, telomere length was 11 kilobase pair, right? 11 kilobase pair, and you have to find how much telomere length is lost in the differentiated cells. Means you have to understand what is the difference between the telomere length in the stem cell and in the differentiated cells. So what you will do, it's easy, right? We'll just simply subtract 11 minus 9.5. What does it come out to be? I think it is 1.5 kbp. So 11 minus 9.5, so it is 1.5 kbp. Okay, so for year 0, the lost, this is the lost length of telomere. Okay, similarly, given the data, you can do it for year 2 as well. This is for year 0, This is for year 2. 10.9 minus 9.4. So how much is the loss? 10.9 minus 9.4. Here also 1.5. Same is observed for year 2 as well.
Yes, ma'am.
Yes. Yes, Janvi was saying something?
Yes, ma'am. 1.5.
Yeah, 1.5, correct. So similarly, you can do it for year 60, where 8 minus 6.5. Here also 1.5.
1.5.
Okay. Now, we won't do it for the year 70 and 80. Why? Because the data given here is not for 30 doublings, rather it is given for 10 and 20 doublings. So from the data given, we can now conclude that after 30 doublings, around 1.5 kilo base pair length of telomere is lost in the differentiated cells. Okay. The next question, what is the rate of telomere loss per doubling? So you have to basically find out the kbp lost per doubling. You have to find that kbp value, this question mark. Okay, so how you will do it? Again, you will use the data. Oh, sorry. Again, you will refer to the data given to you in this figure form. So let's assume for year 0, you know the year 0 data, complete data is given to you. The length of telomere before differentiation means for the stem cell, and the length of telomere for the differentiated cells, that is given to you. That is why I'm choosing year 0. Similarly, you can do this calculation for year 2 as well. So 11 kbp is the length initially, where after 30 doublings, so 30 into, say you assign some variable to the lost number of base pairs. Okay, so variable can be a, gives rise to 9.5. Okay, so this is a type of arithmetic expression that I've written here. You know this value, correct? Do you know it? So if we write this expression in this way, 11 minus 9.5 is equals to 30 into a, and this value is coming to be 1.5 into 30 into a. So you have to just find out this a. A stands for the number of base pair lost, correct? Because if you minus this, then
0.0.
huh? Yeah.
1.05 something.
So you're getting, let me check. You are getting 0.05. Right, you're correct, Janvi. So this 0.05, the unit is kilo base pair. You just have to convert it to base pair, means you have to multiply with 1000. It will come out to be 50 base pair. This will be the value of a. So doubling per doubling you are losing 50 base pairs. All right, so this is done for year 0. Now if you want to confirm this result, you can do it for year 2, because for year 2 this number and this number is given to you, and as well as you know how many doubling, cell doubling this it is undergone, the differentiated cells has undergone. So you can confirm this and see whether you're getting 50 doublings or not. So for year 2, 10.9 minus 30 into a is equals to 9.4. You can simply write 1.5 is equals to 30 into A. So A is basically 50 doublings, 50 base pairs. Alright. So per doubling, 50 base pair loss is there. Now, is it true for the case where the number of doublings is not 30? Is it true there as well? So we can check that.
Yes, madam.
So for year 70, you can check 8 minus 20 into A is equals to 6.5. Okay, and there is a loss of 1.5, 20 into A. So because here number of doublings is 20, right? So let's check. Now I don't think
0.07.
point point zero. How much?
0.075 something.
75. Okay, 75 something. So now, or you can write it as 75 base pairs, right? When you convert multiply with 1000, you get 75 base pair. Now you can just see that at year 70 or 80, the telomere length is gradually decreasing, means it is decreasing at a faster rate, right? Because now the rate is not 50, the rate has changed to 75 base pair, and you can check for 80 as well. Then the answer to this question, you can write that when there is 30 doublings, up to 30 doublings, cell doublings, you are getting a rate of 50, whereas when the doublings is changing like 20 or 10 for year 70 and 80, the rate of loss of telomere is something else like 75. So you can write your answer like this. Now what is the telomere length of the daughter stem cells and differentiated cells after 1 doubling, 3 doublings and 40 doublings? So if you check this graph, what is the telomere length with daughter stems? Okay, so for year 1, this value is missing and this value is also missing. For year 3, same, and for year 50, so it is 50 not 80. Okay, so for year 50, this value is also missing, so you have to find the question marked values. How you will do that? Now see that for year 1, 3, 50, the number of cell doublings is what? It is 30 only, correct? It is 30, and for 30 cell doublings, you know the rate of loss of telomere length is 50 base pair per doubling. So now you can now again frame an expression for you. Now will you frame an expression? So from this side, you don't know the value to start with, minus 50 doublings into 50 doublings means 50 base pairs is being lost per 30 for 30 doublings, and you have to find this value. Okay, is this expression clear? This x y, this expression, is it clear? Now what will be the value of there are two variables in this expression, correct? You cannot determine without finding, there's only one expression but there is two variables, so you need another expression. So how will you do it? Any guesses? Any guesses? So there is, as far as I can think of, you cannot solve this expression. This expression is completely moot. Okay, you cannot do it. Rather, what you can do is choose a value of x as 11 kbp. Okay, why 11 kbp? Because it is for the year 0. All right, initial one. And here, when you are coming to year 1, okay, just give me 1 second. Okay. Okay, so we can actually do it. Sorry for like taking some time. We can actually do it. We can I actually forgot about the assumption that I told you about, that there is no difference in the rate of telomere loss between the stem cell and the committed cells. Okay, this means what? This means we can uh, get a value of x. How? So for year 0, the telomere length of the cells were 11 kbp. Now the stem cell clone that is formed in year 1, if we assume that the rate of loss of telomere length is 50, then what will be the length of this telomeres in year 1? It will be around 11 minus 0.05, 10.95 kbp. Okay, then this will be value of x, 10.95 kbp. This is base pair. Okay, 50 base pairs. So now you have the value of x with you. So now that is straightforward. You can easily, so this is the like base pair. It is better to use in kilo base pair, means 0.05. Okay, don't, means all should be expressed in kilo base pair for uniformity in the expression. So into 30 is equals to y. So if we just minus this, what you will get, so y is coming out to be 9.45 kilo base pair. Okay, so that will be the so x is the length of the telomere of the stem cells in year 1, and for the differentiated cells that is coming after 30 base pairs, after 30 doublings, this will be the length for them. Similarly, you can do it for year 3 as well. Okay, then you can do it for year 4, year 5. Okay, so in this way you can do it. Now there is year 50. It is, how will you do for year 50? Maybe you should think a bit, but I don't think it should be that difficult. You can do it. Okay, take your time in like doing it on your own for the other two doublings, year 5, so year 3 and year 40. Okay, just do it on your own. Now for question 4, what it is written, that it is assumed that cell senescence occurs after 90 doublings. After 90 doublings, the cell undergoes the senescence stage, means it has become very old, and after which the cell will start dying. So what is the telomere length after 90 doublings? Okay, so you have to find out what is the telomere length after 90 doublings. So y is equals to 90. You have to do it. So first you have to find the value of A, the rate of telomere loss for year y is equals to 80, because the data is given to you, right? And use that A value to determine what will be the value of x, means the telomere length of this stem cell at the year 90. And from that you can get the value of y, that is the telomere length of the differentiated cells. Okay, if you do that, if you get the values of a, x and y, then and write it in the same columns as it is there in the question above, then you can simply just minus it, or you can actually the answer will be y only. Yeah, so answer will be y. The question, so I can do it for you. So let's see. So for year 80, what will be the value of a? A will be equals to 1.5. So A was coming out to be 75 base pair. No, it is 10. Sorry. A will be 15 base pair. All right. 0.15. Oh, sorry, 150 base pair. 2000. Okay. A will be 150 base pairs. That is A. That is the rate of telomere loss. Okay. Now you assume that for the year 90, for year 90, what is happening? The stem cell clones will have a telomere length of 8 minus 0.15. That is 7.85 kilobase pair. So this is the value of x. So you have to determine what is the telomere length of the 90 doublings. So you have to determine the value of y. So if for 90 doublings, no, okay, fine. The number of doublings, say it is going 10 only. The 7.85 minus 10 into 0.15, this will be y. So solve for y, you will get the telomere length. Okay. So now you can do a similar kind of questions if it is given to you during the quiz. Now few more topics. So in this slide, you will understand that how the stem cells are important for functioning of a tissue. Okay, means for maintaining the dynamic state of the tissue, for ensuring that the cells in this tissue undergoes the cellular fate processes, etc. So in this table, here in this table is given for the hematopoietic stem cells to show you how the stem cells actually form the differentiated cells through undergoing stages of differentiation. Okay, and what is the characteristic of each stage? So it is observed that first the stem cell differentiates and enters a stage of early progenitor cell, or it is also called pre-progenitor cell, I think. Yeah, pre-progenitor cell. The characteristic of this cell is it replicates very slowly. After it leaves this stage, it enters another stage that is the progenitor cell or the late progenitor cell. Now here the rate of cell division is very high, but also the apoptosis rate is also very high. So both these rates are very high. So because of which at this stage the importance of the survival factors, survival growth factors becomes very important, because if they are not present, then whatever new cells are being formed, they will undergo apoptosis. Okay, so at this stage you need these survival cells, or you need to supply the survival factors to permit the progenitor cells to enter the next stage of differentiation, that is precursor cells. Okay, now these precursor cells, they again the rate of replication, it becomes slow. Okay, there is a slow rate of replication, and this precursor cells is now ready to become the differentiated cells with all the necessary morphological features of the differentiated cells. Like for the hematopoietic stem cells, the blood cells will be formed, and as soon as they are formed, they leave the bone marrow and enter the blood circulation to perform their intended specialized function. So this table shows you how the cell number varies for each of the stages. Then cell cycling rate, like I told you, the pre-progenitor cell, the cycling is very slow, whereas the progenitor cell, the cycling is very fast. Apoptosis, how it is going on, then motility. So the precursor cells has a very high motility, because at this stage they are kind of ready to leave the bone marrow, because immediately of the precursor cell, mature cell is formed, correct? And how the regulation from moving from one stage to another is taken care of. So for stem cell, it is majorly the cell contact that determines the activation of the stem cell. Then also it is true for pre-progenitor cells, whereas for the late progenitor cells, or the progenitor cells or precursor cells, there is a soluble factors. The presence of soluble factors regulates the transition from one state to another. So that's it for today's class. It almost went on for 2 hours, and but I could finish this small chapter actually. And if you have any query, you can let me know, and please go through this question on your own once and try to understand what all different types of questions can come out of it. Okay, and try to solve it. Various variations of questions, you can frame it yourself from this figure and try to solve, and this way you will be able to understand this topic better. And yes, is there any query? Let me know now while I prepare for your attendance. Meanwhile, I am stopped sharing. I have stopped sharing. Okay, meanwhile I have stopped sharing, and if there is any query, you can let me know now. Okay, so let me know who all are present today. Janvi, Vaishnavi, you guys are present, right? Present in front of the screen?
Yes, ma'am.
Okay. Janvi, Vaishnavi, then Vani is there.
Yes.
So Kirtan texted me saying his microphone is not working, but he is present. Okay, so I'm marking Kirtan as present, and Shambhavi is also present, right?
Yes, ma'am.
Okay. So 1, 2, 3, 4, 5. 5 of you are here. So if there is any queries, let me know. I have marked your attendance for today's class. I'll also share the recording as usual and the slides so that you can read on your own, and let me know if any further queries come up. Okay. So recording is being, let me stop the recording.